bilateral, or the bullet-shaped / conical that HIV has. It's the ones where we don't know what to do with this, like bacteriophage is the other toxin, okay, complex. And so this is smallpox virus, and so this is going to cause smallpox, which is, you know, fatal. And maybe if you can stomach it, you can look up what that looks like to get smallpox. Like big, huge pustules, like blister-like pustules, like all over the body will, you know, burst and leave a permanent scarring. But that's just a window as to what's going on the inside too. This is basically after epithelia, which you have pretty much everywhere. So it leads to the destruction of tissues. You can't survive this. Okay. There are survivors of smallpox. It's a mild condition. People who are, you know, kept away from everybody else and worked through, you know, I don't know, various ways. I don't know if I ever had to heal somebody from smallpox. This is hopefully an ancient disease, but who knows anymore. So, all right, very few people are still alive that have been vaccinated. So that's far. Okay, but now we're on herpes. And herpes virus, herpes viridae, is a family of virus, and there's lots of them in here. And so you can choose whichever one you want within this grouping. So it includes herpes simplex, and I told you the other day that herpes simplex type 1 and type 2 are oral and genital, and it used to be that 1 was always oral and 2 was always genital, but now they find the oral in the genitalia and the genital one in the oral cavity, so it's like this. Right, so it's after those kinds of epithelia. Then we have varicella virus, and that is chickenpox. So chickenpox and herpes simplex are in the same family. So they are double-stranded DNA viruses that are enveloped. Okay, they have that in common with smallpox, but their capsid is different. You see that? Okay. And so they also include a couple others that are worth describing. One of them is the Kaposi sarcoma. So Kaposi sarcoma is a very mild virus, and most people have been exposed but didn't get any kind of symptom. And Kaposi sarcoma causes like small, it kind of looks like a bruise, like a purpley bruise. It begins on the trunk and it spreads upwards to the face. And typically, if your patient presents with this, it's a clue that this person has an immunocompromised situation going on, typically AIDS. So that's one of the first signs, is having a Kaposi sarcoma. Very strange and rare thing, but it's indicative of not having an immune system, essentially. Okay. All right, so then we're gonna march through the hepatitis viruses. All right. So the hepatitis viruses are all very different from one another, but they're all named hepatitis virus because they do the same thing. They're after hepatocytes, and they'll cause an inflammatory response in the liver and all sorts of issues beyond that. But their transmission and what nucleic acids they have and whether they're naked or not, that all differs. So the first one is Hep B. If you're going into healthcare and if you work there now, you maybe have already been vaccinated against hepatitis B. It's a 2-part vaccination, and that's because it's transmitted through the blood. And so choosing to go into healthcare means your accidental needle sticks are part of your life. So you have to be protected against it, and typically you can't even be hired until you have the series. Yes?
Now they give all newborns Hep B first day of life.
Now they're doing that?
Yeah. The vaccine.
They give them an adult dosage?
I don't know about that, but it's
Yeah.
included with the vitamin K in the treatment.
Okay. All right. We'll talk about vaccines later after we get through immunology. All right. So that's Hep B. Now that's it for the DNAs that I have on this sheet. Okay. There's more than this. All right. So now we're in RNAs, and we are looking first at single-stranded positive sense. What does it mean if it's positive sense? The start codon at the 5' end so that it's ready to be translated right off the bat. That's what that means, positive sense. So it's not about charge or anything like that, right? So it's just positive sense is what they call it. And this first one is a very small virus, these enteroviruses, and it does include polio. So polio, very, very tiny virus, but it's in the same category, the same family as rhinovirus, which is the common cold. That's not scary. That one's really easy. But here's another one, Hep A. So do you see that Hep A and Hep A are nothing at all alike? One is double-stranded DNA, one is a single strand of positive sense RNA, one is enveloped, one is naked. Okay, they're not the same. They just have the same target cell, not the same receptor that they land on, but same organ system. Okay, Hep E is a calciviridae family, and in it is also norovirus. Norovirus is, I meant to show this picture. I took a picture, see my phone is full of so much dumb stuff, but it's all from class, about the, there was, let's see, it was on the cruise ship, it just recently happened. Again, norovirus. So, it happens a lot, I know, I'm sorry. I don't know, anyway, I had a picture of it, I might include it somewhere if I can find it. So, norovirus, highly contagious gastrointestinal virus, and it's so tiny, and almost impossible to be found. It's just something to think about. Most people are gonna get over it, but very old, very young, they have a problem with this. Okay. All right, then rubella. So you've heard of measles, mumps, rubella. Rubella is an RNA virus. And it's positive sense, and this one's enveloped. But it also includes in this grouping, Togaviridae, that's the picture that I had that was an electron micrograph that showed you the receptor-mediated endocytosis, right? Like that coming in, that was the Togaviridae. And so it also includes Eastern and Western equine encephalitis. So if you have a horse or around horses, they have to get vaccinated against this. Okay. Then we have Hep C. Hep C is a positive sense RNA virus enveloped, smaller than these rubella virus. But this one includes West Nile virus and also yellow fever. So if you go, somewhere tropical, they might ask you to get a yellow fever vaccination, and it's because you get this transmitted from mosquito bites. Also, dengue is in that same group. And then here we come to coronavirus. Now, this is an older book, and it was written, I've been teaching this, as I said, 20-something years, and I'm not on the same edition or anything. But the original coronavirus what came out was really like 2003. And that was really scary because it was young people who were dying, which is unusual. Typically, respiratory ailments, pneumonias like that, that usually is what afflicts older people, really old people, especially like something has to take them out at the end, and very often it's pneumonia. And so they were surprised that here's this coronavirus that was killing young people. And so a lot of work went into vaccines. So then, 2019 comes around, and here's SARS, again, similar, but it wasn't the same, but it was in the same family. And this is SARS-CoV-2. So this is the one that causes COVID. Okay, and so we're more familiar with this simply because it was in the news all the time, but it too is a positive sense RNA virus. And so it has an AUG at the start site, right? And it has a spike protein. That spike, that's what all the antibodies, that all the vaccines were designed to give you antibodies to the spike. If we block the spikes, it can't land on the cell, and if it can't land on the cell, it can't get in. And because it has no metabolism, if it can't get in, it's game over for them, right? So it's a wonderful way to go about that, that's fine. This is actually a helical virus. You can't quite see that. They're showing you the RNA here, but this is like one of the capsimeres that will be going all the way around it in a helical format. Okay, so it's a helical. Most people get over it, but much worse for immunocompromised. And we can talk about a little bit more about that in a little bit. Okay. Now, negative sense. include things like the rabies virus and so this is negative sense enveloped. Rabies virus that Pasteur was working on. And then I'm gonna jump down to Hep D. So negative sense, just one strand of RNA, but if it's negative sense, it has to bring with it replicase. We know that. Brings it with it, as does rabies. And then it's hepatitis D. Okay, so at this point we have seen A, B, C, D, E, right? And they're all different. So I'm just saying, if you were filling out in your table, you could just choose all hepatitis and be able to do them all. I'm serious. Right? Like, okay. All right. And then the last one on here is influenza. Negative sense. Okay, but this one has multiple strands, up to 8 different strands. And this makes me want to go back here for just a moment, because I really need to say this, and thankfully I'm on PowerPoint and not Canvas where I can do this. Replicase. No proofreading. Now you might remember back when we were I at least was going over, should I talk about DNA? What do you really need to know? And I'm kind of regretting that decision, to be honest. So just count yourself lucky, because fall of 2025 is getting a full dosage. But replicase is an RNA-dependent RNA polymerase. It has to make all RNA viruses need it to make copies of their RNA, because the cells don't do that. Cells make RNA from DNA. Okay, fine. It doesn't proofread, which means it's making mistakes. And when we make mistakes, those are mutations. And those mutations, what that means is you're going to be making like missense mutations, which means that you're going to have different expressed spikes, because that's where the information comes from. It is from the RNA, right? So if the spike is constantly changing, then the antibody you made to that spike is no longer useful, right? Okay, so thinking along those lines, here is SARS-CoV-2. It's an RNA virus. I knew right away, okay, this is replicase, this is making mistakes. And when they said, oh, there's the Lambda variant and there's the Omicron variant, I'm like, like myself and every other microbiologist was like, yeah, it's an what do you want? It's replicase. Like it doesn't even care what it's doing. It's so sloppy. Remember even transduction, how sloppy it was about like just shove something in the capsid and let's go, right? I mean, it just like does not care. So okay, that's SARS-CoV-2, one strand of RNA using replicase to make the copies. And then here you're looking at the flu, 8 strands of RNA using replicase to make the copies. So the likelihood of mutation in both is high. It's 8 times worse in the flu. The flu absolutely is moving in a different direction all the time, because of, this is what happens with replicase. So is SARS-CoV-2, but not at the same rate. Okay? So, and the other thing is, and you know this too now, who is the least to the most resistant, at the very bottom are enveloped virus. And that's what this is, SARS-CoV-2. So when everybody else was freaking out, I'm not saying I wasn't scared. Okay, my daughter was in Munich at the time. It was a little freaky for me. But I was like, well, okay, yes, it will change. But we can clean this fairly easily, because it's at the bottom of my list. Okay, it's not like I'm trying to fight this really bad endospore, right? Like that, then you're like, oh boy, right? But this is, okay, all right. And then the last one here that I wanted to point out was, 3 more, sorry. Okay, hantavirus, right here, negative sense, single-strand RNA enveloped hantavirus. Why am I bringing this up? Because a few years ago, I think it was like I'll just throw it out a number, 2012. They closed down Curry Village in Yosemite because hantavirus is spread from inhaling the feces of rodents. So little mice go under the Curry Village doesn't, it's like tent cabins, but they're on a platform, and the platforms have little slats between them, and the little rodents and critters go underneath to keep warm, and of course they, defecate, and then that dries and would come up through the slats, and people were just breathing that all night, and so then getting this respiratory symptoms, and bad if you're in bad shape. Okay, HIV we will spend a lot of time on. This is a retrovirus, and I'll describe that. HIV is an RNA virus that has 2 strands of RNA, but it converts it to DNA, and we will describe this in detail. Yes? So that if you're hemorrhaging, you're bleeding. So that can happen for some people, yeah.
That sounds horrible.
Oh, it is horrible. It is horrible, yeah. I mean, for most people, they don't feel well and then they get over it. But then for some people, that's what happens. Okay.
And could that be fatal?
Oh, yes.
I was gonna say, like bleeding in the brain,
It's not even so much in the brain, it's in the lung. We can't take, so there's only so much space in the lung for gas exchange. And we have this little problem. You took your biology, right? Okay, so like if you were a bird, then you would, your lungs are like a bellows, and underneath there's something to push out even all the old used up air. We don't have that, so you have this little like layer of air that you can't quite get out, and it's CO2. So that if it's there, then that's, reduces the amount you can bring in. Okay, that's just how it goes. If you have some sort of pulmonary situation like this, bleeding, inflammation, or bleeding, you are reducing the amount of lung space, which means you have reduced oxygen, which means, and we now know this, less ATP to run your immune system, which we will come to find is really ATP heavy. You want it to run well, it's got to have it all. It's got to have it all, which means you should go to bed when you're sick. Go do nothing and let that work. Okay, not go out. I'm having a conversation with my son right now. Yes.
For the table itself, do we want the virus name?
Yes.
Or the viral name?
You can go with name, because suppose you chose, and I'm okay with chickenpox today. Okay, so if you chose the family, then you would just have to be stuck by saying herpes. Okay. So what I want is something that will stick in your head. All right, and that even your poor spelling, I can figure out what you want. Okay?
Thank you.
I really do try with some people, but sometimes I'm like, I just big question mark, I don't know what you're trying to say. Okay.
On that table, do you also want us to state if it's double-stranded or single-stranded or negative sense?
In my heart of hearts, I do want that, but I never get it, so I'm not gonna hold you. I won't hold you. Okay. All right, so to make more virus to study, it has to be grown inside of a living cell. And so you can use tissue culture, and I did this for years, not with virus, but for different reasons, and I grew tissue. So epithelia will grow, in fact, on a plate, given the right conditions, etc. So if you could take epithelia that's growing well and then inoculate it with virus, you would get more. Okay, virus cannot propagate on their own. That's completely unlike bacteria, where you can just take a loopful, put it on a plate, it'll go. Take a loopful, put it in a broth tube, it'll go. You can't do that with virus. Broth, it doesn't matter what's in it, they can't make use of it. There's no cell in there for them to contribute to, right? That's the big difference. So that's the problem why we can't work with virus in room 317, is because the first thing we would have to do is learn tissue culture. So that would be, that would be like level 2 micro. Yes?
Our slides are not matching up. with
Okay, so
your
maybe you
slide.
missed the first part of this conversation where I said that. This is the same slide.
Yes, yes.
Yes. The next one has an egg too.
Yeah.
Okay. Yeah. I mean, it's all the same information. I think this is better. Okay. So if you want to grow bacteriophage, then you have to grow it onto bacteria. So you make a lawn, and then you squirt it with what you think might contain the phage virus, and if it's there, they'll go in, they'll climb inside the bacteria on that plate and destroy them. And everywhere that you see these little holes, they call them plaques, that means that was a cell that burst. And so that's your evidence that if in fact there's a virus present on the plate. Okay? They cannot grow in a test tube. Okay? They can't grow on a plate, and we know why. right? So if I were to ask that question, you could explain why that is. Okay. So, animal viruses are always have to be grown in an animal cell. Okay. Eggs make a great place for that because first off, they're inexpensive. Number two, the egg immediately gives you both big cytoplasm, that's the white part, and then the nucleus, that's the yolk, right? So we'll see that DNA viruses are going to require all of the machinery to copy DNA that exists in the nucleus. RNA viruses need, well, they have to bring their own replicase, but they, either way, they don't need to climb inside the nucleus, they're gonna be in the cytoplasm, so you have plenty of cytoplasm to work with here too, okay? So sometimes if you're watching the news and they say something about like the new batch of flu vaccine is available now or something, you'll see a bunch of eggs going by in the background and you're just like, wait, that's this. So what that also means though is that if you have an egg allergy, you have problems with a lot of these vaccines because they were in fact made inside eggs. Right? So it's just something to think about. So virus can be grown in cell culture as well, as I just said. You take cells, you put them on a plate, they will in fact grow. Epithelial cells, almost the definition of them is like almost evenly sized cells growing in a sheet, right? If there's virus in one of these cells, then what sometimes happens is that instead of growing into even sized cells, and they know when they've hit the other cell because of contact inhibition with their surface sugars, we've talked about this, yes? Okay, one cell knows when to stop. Those that have virus, they don't care, and they continue to divide over and beyond their borders, and so they'll form a little tumor. So that's how some viruses react, but there's these cytopathic effects, and this is what you don't have, is sort of an explanatory title on that page. So I will posit to you, Leah, that this is the superior. So cytopathic effects. This is just a way to figure out which virus is it. So as the clinician, you take the sample from your patient, right? They come in, I don't know, I feel terrible, blah, blah, blah. Okay, so you take the sample from wherever it is. It is from their blood, their sputum, their stool, whatever it might be, okay? And now you send it off to the lab, and they'll try to do a gram stain. But if it's a virus, then that's not gonna work, right? You can't see anything. So what they do is they put it onto tissue, and then they check for the cytopathic effects. Cyto means cell, and pathology is what happens to it, like the bad thing that happens to it. By putting the virus on cells, they check to see what happened to the cell. And you will see that for each one of these, there is a different effect. I will ask you for one virus and its cytopathic effect. That's what I mean. This slide here, slide 41, on this number in particular. Could be a different number for you, Leah, don't know. Okay. So if it's smallpox, the cells, now remember it's epithelia, so they should be like squamous, like flat tile, okay? Instead they'll round up if there's smallpox in there, they round up, pull away from each other, okay? And you'll get little spots to appear in the cytoplasm. They don't even try to figure out what those spots are, they just, okay, boom, that means smallpox is here. They see this happen. Cells round up, inclusions in the cytoplasm. With herpes, it's different. The cells, instead of, they're all like squamous, flat, they all fuse into one giant cell. And then it looks like you're looking at a ton of cells, or one giant cell with lots of nuclei. So they call it multinucleated syncytia, but basically it's fused cells. That's a very different response than what just happened with the cells inoculated with smallpox. Okay, so you would be able to know. Adenovirus, that just causes a common cold, but the cells clump, and you get inclusions in the nucleus. Polio, the cells lyse. That's usually what students choose because it's easy to remember the cells lyse. Okay, I don't have to worry about inclusions here or there. Okay, whatever. Polio is also an RNA naked virus, so it's like an easy thing. You just make one thing. All right. How about measles? Syncytial as well. Okay, but no nuclear inclusions, as you see with herpes. Measles, unfortunately, I think you guys had best start learning a little bit more about it. Okay, and we'll talk a little bit more about it. All right, so normal cells, a nice even sheet, but then you're gonna get different things to form if they get inoculated with virus, including this. This is what happens with herpes. Instead of being these individual cells like that, they're just one big clump. Okay, so that tells you right away there's something wrong in here. Okay, and it just so happens to be they're virally infected. Okay, so we know the general characteristics of virus. We recognize that virus are super diverse in terms of their type of nucleic acid and whether they are naked or enveloped and what kind of capsid they have. All of this is what's going to put virus in different categories. Okay, but now they have to replicate. So how do they do so? And you'll notice I'm not saying reproduce because they can't do that. Okay, but that more copies of them in fact are going to be made. So here's the steps. Okay, they're going to absorb and penetrate. Okay, so they're going to. We already know all. Like you could tell me right now how it is. How is it that it knows which tissue it wants to go after? Which cell it wants to absorb to? That's the receptor, and what is it? That's on the tissue. But what does the virus have?
Spike.
A spike. So the spike lands on a receptor, and then penetration. What do I mean by that? Like how it actually gets inside the cell. Exactly. Receptor-mediated endocytosis, fusion, or injection if it's talking about phage. Okay, uncoating. That part we've not described because the name of the game is the nucleic acid. They've got to get the coat off. So we'll describe that a little bit. And then now protein synthesis. So they need to have all of their proteins made. What kind of proteins would a virus need? A virus is in fact genetic material in that way.
Enzymes.
So, okay, enzymes for like replicase would be one. That could be a protein. But what if it's a DNA virus? It doesn't need replicase. So what kind of protein might it have? Like how did it land here in the first place? The spike. Spike is a protein. Okay. The capsid is a protein, right? I'm telling you, it's a secret answer. It really is. Everything's a protein. That's how we work. Okay. So they'll have to get the cell to do the protein synthesis. And then maturation, this is self-assembly. Okay, this is they put themselves together. And then they get released. And so that part depends. If it's enveloped, it's gonna bud off. That's pinch off and take a little bit of plasma membrane with it. Okay, not the entire membrane, just a little bit with it, right? With it. But if it's naked, what's gonna happen? It's just gonna burst, that's right. Okay, so these are the general steps, and when you're looking at viral replication, it usually causes this one-step growth curve is what they call it. So that there'd be a certain number of virus in the sputum, in the stool, in the respiratory tract, whatever it is, and then it would seem that there's none. This is called the eclipse period. This is when it's inside of the cell. During which time that cell has now become a virus-making factory, making more copies of the nucleic acid, making more copies of the capsid and the spike, okay? And then the virus is released, either by bursting or budding, and now the numbers of virus climb. We get to kind of a peak, and this is what they term acute infection, which means that all of the signs and symptoms that you would associate with this disorder would be evident here. So if it's the high fever, here's where it's the highest. If it's the rash, this is the day you get the rash, like that, like that. But then you see how we clear it very quickly, okay? And the reason for the quick clearing on most viral infections, almost all of them are acute, is because of the cell that is infected. And most virus are after epithelia. Epithelia is mitotic. We slough it, okay? Whether it's on the skin or inside of us, that's what we do. That's what we do. Now, how quickly do you do that? Well, okay, how old are you? Are you well rested? Are you eating well? There's like a lot of thoughts in here, right? So if you consider the 3-year-old coughing, sneezing, sick, gets grandma sick, like the 3-year-old, like a day and a half later, you're not even sure that ever happened to that kid at all, and grandma is still sick with that because the replacement of those cells isn't as fast. That's what happens. So, okay. I found this online, and this was actually a little animation, so I was like screenshotting different pictures, so let's see how this works. So same picture, certain number, then goes down, okay? So that at first there's a certain number you can detect and they're not inside yet, okay? That's the attachment phase. Then they penetrate, okay, now it seems like they're gone because they're inside. Okay, seems like the number is 0, or close to 0, okay? All right. Then in here, now they're getting uncoated. I'll describe this, okay? Getting uncoated. Then biosynthesis, which means they're tricking the cell into making all of their stuff. Then they mature, okay, we're still here. And now we're bursting out, boom. Okay, so all of this period here that seems like nothing's going on is they're actually inside the cell. That's when they're gonna burst out. And do you remember what I told you, how many you can expect? Thousands. So this value goes from 0 to a lot in that part. Okay. Most viral infections are acute, cleared by the immune system, and we will remind ourselves about the lytic and lysogenic cycle here because it plays a big role. This picture is better in the sense that you get to see all the pieces and parts that have to be made for phage, because it's not as cut and dry as the next picture, but I actually prefer the next picture for different reasons. But here you're just seeing that there's actually a head and then this sheath. Okay, that's what acts like the syringe. It's got tail fibers. It's very complicated structure. But so this is the one we're more used to. So that's this very simple picture is just trimmed down version of the last one. Okay, so this is the phage landing on bacteria. This is the lytic cycle. This is the lysogenic cycle. We talked about some of this already. So the phage lands on the bacteria and uses the shaft to act like a syringe to squirt its DNA in. Bacteriophage is a DNA virus. So it comes in and it circularizes, and now it forces this cell to become a virus-making factory. It takes over the cell. It uses the cell's machinery to make copies of the DNA. Well, what would that be? That would be the helicase, the topoisomerase, the DNA polymerase. All of those things would be utilized to make more DNA. It would use the cell's ribosomes to make all of the capsid. Okay, and in ways we don't understand, they come together on their own, and then it bursts out of the cell. That's lytic. But the lysogenic, okay, those phage lands, squirts in the DNA. The DNA is circularized, but sometimes the DNA of the virus becomes part of the chromosome, and now it's called a prophage. When this is the case, when this cell now divides, the first step is in binary fission is copy the DNA. It would copy the DNA. Both cells, both daughter cells now have secretly, they're carrying viral DNA. And this can go on for many, many cell divisions until we get to the arrow, bless you, which I told you before to just write stress, right? Okay. When this is stressful for the bacterium, and we know when that would be, they have temperature optimums, they have pH optimums, right? Osmotic pressure optimums, whether there's enough nitrogen and carbon, all of those things have to be in their favor. Otherwise, the virus is like, I don't want to be in here anymore. I'm out. So it excises itself from the chromosome and undergoes the lytic cycle and releases. But now instead of one infected cell, it could be a ton. You know, if this were E. coli, 17 minutes later, you're looking at a set of 2. And then another 17 to 20 minutes later, it's 4, right? So it could be quite a few launching thousands all at once just because of some stressful event, okay? For ourselves, this is the acute phase. This is going to be the latent phase. Acute disorders are the typical ones. You know, even the flu, the common cold, these things. Okay, you get sick, and then you turn into a virus making factory. You get rid of a bunch. You get other people sick too. Okay, all right, and then it just like clears. All right, fine. But sometimes you get things like herpes. and now you're stuck. And so your cell is secretly carrying herpes viral DNA, and then you get stressed, and then the virus is like, oh, I don't want to be in here. Okay? You stink right now. And so then they lyse the cells, epithelia, on your mouth. There's your culture. And you see somebody with that, or you have it yourself, it's shedding. It's shedding right now. That is what it's doing. Okay, that's what that means. And so we know this. Most viral infections, acute, but some are going to go this direction. This is not the exact same format. My cells are much more complicated than bacteria, okay? But this is an excellent model for what's happening, okay? Does that make sense? Okay. All right, so we know entry requires attachment with a spike, okay? If it's naked, it gets brought in like this, okay, and that's receptor mediated. But if it's enveloped, here's herpes, spike attachment, here's the plasma membrane of your cell. This is an electron micrograph, of course, and then we get the fusion such that the lipids on the envelope stay on the outside. You see how it never even got in? Okay, this is, they have radio labeled it probably. I don't know what they did. Typically that's what they do. And then so that you can see, oh, that never even got in. Okay, so basically it doesn't need that membrane for any other reason.
The envelope.
The
Stays.
envelope.
Okay.
Yep, stays on the outside. It just fuses and kind of dumps the contents. All right. So attachment, different viruses are going to have different attachment mechanisms. Okay. And because of that, they're going to land on different tissue types. That's tissue tropism. We already said that before. So I'm kind of going back and forth review. Okay. A little bit. All right. So we're clear on that. Once it's brought in either receptor mediated or by fusion, and that's what this is supposed to show you, okay? And now the contents are dumped in here. What we're not seeing here, but we know that when a cell invaginates like this and causes or forms an endosome, the very next step would be the lysosome would come, right? And fuse and dump its contents, and the lysosome has hydrolytic enzymes that cut stuff up. Well, you're helping it. You're saying, can I take your coat off for you? That's what you just did. Okay, so they're really well suited to coming into a cell. Okay, take that coat off. So here it's a little bit different for the envelope. They fuse, they dump in. Here is their capsid, but they themselves will disassemble. Okay, and release the nucleic acid. Okay, it's the nucleic acid which has to come next. So here it is, the lysosomes coming in, but I don't actually have a picture of that. All right, but now here's where we kind of have to make a fork in the road, because if you're a DNA virus, you have a different story than an RNA virus. And so I'll spend a few moments on this because this is an important virus, and I absolutely will ask every bit of this on the exam. Okay. So this is HPV, human papillomavirus. HPV is a naked DNA virus, okay? And HPV, the spikes on it land on basal epithelia. Okay, so that's the lower layer. You've seen pictures of skin before. The top layer of epithelia on your skin is dry and dead and sloughs, right? Okay, and we said that's an excellent barrier against virus because they can land on you all they want. Those cells are dead, right? You're going to slough those anyway. But if you scrape yourself, okay, or you've had some other abrasion, that top layer of dead cells is gone and you've exposed the actual basal epithelia. That's the stuff that's dividing, right? Okay, then that's what they bind to. So any kind of abrasion is going to expose the basal epithelia. So once that binds to the basal epithelia, it is going to, because it's a naked, how's it getting in? Receptor-mediated endocytosis, okay? Not shown, okay? But you would have the vesicle made. I have a picture of it before, this picture here, okay? The vesicle forms, it brings it in. And what I don't have here is the lysosome, right? Comes over and helps uncode it. So here you see the capsid proteins. And here's the DNA. Now, what is not shown in this image is here's DNA, here's the nucleus. Okay, this is your cell, your epithelia. Suddenly, the DNA is inside the nucleus, which has got to give you pause. I hope it does, because you know that the nucleus has not one but two phospholipid bilayers and pores that only allow certain things in and out. Okay, the only thing that gets to come out is message, and it has to have a bunch of stuff added to it. I skipped all that for you, okay? But like, you can't just come out, okay? And stuff can't just come in. It has to have a nuclear signal, okay? A certain sequence on the protein to get in. But remember, what is the target cell? What cell did it infect? Basal epithelia. And epithelial cells is they're sometimes called mitotic tissue. Have you heard this before? Which means they're doing what? Mitosis. Okay, so we'll have a little teeny 3-sentence story about mitosis, because you know how mitosis goes. In fact, you know you're in mitosis when the nuclear envelope is disassembled. Yes? That happens in late prophase. And that has to happen because that's how the microtubules can grab the sister chromatid and pull them to the poles, right? Yeah? So there's a section of time in the life of an epithelial cell when the nuclear envelope is in fact broken apart. But by telophase, it shuts back down, right? Okay, now it's inside. So now it's inside the nucleus. And inside the nucleus, it's using all of the nuclear machinery. So RNA polymerase lives in the nucleus because RNA polymerase makes a complementary messenger RNA copy from DNA, right? From genes. DNA lives in the nucleus. RNA polymerase is one of those proteins that is brought into the nucleus. Okay, so it makes message. Okay, it's using my RNA polymerase, and it used my A's, T's, U, and G, like all of that. Not A, C, and G, right? To make the message. All of that. Message is one of the few things that gets out. Okay, that goes out here. The ribosomes, not shown, are the ones that are going to translate that message into capsid, into spike, right? Okay. Meanwhile, my helicase, DNA polymerase, topoisomerase is busy making copies of the viral DNA. Okay? These capsid proteins, which are made out here, actually have a nuclear localization signal. That just means they have a certain sequence as part of their amino acid sequence that allows them back into the nucleus. Okay? There are proteins in the nucleus, RNA polymerase, helicase, topoisomerase, those are all proteins. They all are made out in the cytoplasm, but they get shuttled in. Okay? So the capsid proteins get shuttled in, it self-assembles. How? Can't tell you. I don't know. Okay, the cell we know is not equipped for this, and we know that if we have capsid and nucleic acid, you come back later, they're together. We do not know what the heck's going on. There must be some attraction. That's what we say when we have no idea. Okay, there's something going on. So it self-assembles and it leaves, but this is showing it all nice. How does it really leave? It's naked. Oh, it bursts. It bursts. Now, most human papilloma virus is making warts. That's it. These burst cells that you have are essentially going to be little holes in your epithelial layer, which you're going to fix with collagen. Collagen and collagen, you know, but we actually do more keratin on the skin. So keratin is called hyperkeratinosis, and that's basically what a wart is. Very thick skin in this one area where we've just had a lot of damage. Okay? Unfortunately, if you have warts and around them, you know they get bigger because they're just like, okay, I'm done with these guys. Let's go check out things. That's what happens. But remember how it all starts with the exposure of the basal epithelia. So what I'm saying is never get hurt. No, I'm kidding. I mean, I don't know. I mean, it's like, you know, this is really difficult. But That's just part and parcel of being alive, is you're gonna have little breaks here and there, but that's what it's after. Your normal skin that is dry, dead, and sloughs, that's not a good cell for them to target, right? It's dead, can't do anything, okay? So that's kind of a long story. I just spent X number of minutes on one slide. This is for sure a page on your exam, okay? It's not worth a whole lot, maybe like 5 points or something, but it's like, they're slam dunks if you know the story. It's just a story, okay? All right. We're gonna move on to the next one. So that's a DNA virus story, and I chose to tell the story of HPV, but I could have told the story of herpes and it would be the same. I could have told the stories of parvo and it would have been the same. Okay, it needs to get that DNA into the nucleus because that's where the machinery is to make copy of DNA. Yes? That does not exist in the cytoplasm. If you're an RNA virus, you have a big issue. And that is that the cell does not copy RNA. The cell makes RNA from DNA. So you have to bring with you either the information for how to make a replicase or have brought the replicase already, okay? So that's the difference between being positive sense and negative sense RNA virus, okay? So in this image, they both seem to me to be just naked, right? I don't see any envelope. And they're not really showing you what the entry is, but you're supposed to know it. It's receptor-mediated endocytosis. The lysosome would come, it would get uncoded. Okay, here we go. If you are positive sense, then that's all you need because at the 5' end, you're ready to go. That little circle is supposed to mean there's an AUG there, and so we're ready to go. Here comes my small subunit, here comes the tRNA, here comes the large subunit, and it begins translation. Of the many proteins that will be translated, replicase is one of them. Okay, replicase is the protein right here. RNA-dependent RNA polymerase, that's the real name. We like to call it replicase, it's a lot easier. And replicase will make copies of the RNA. First it makes a copy, the complementary copy will be negative sense, and it uses that complementary negative sense to make many, many copies so that they're all positive sense. In the meantime, all the other proteins are being made by your ribosome, yes, with your amino acids, yes, and this is an anabolic process that takes ATP. This is your ATP. You had special plans for that ATP. Too bad. You're being taken over by these guys, and so now this self-assembles the RNA inside the capsid and it is going to burst out because this is naked. If it were enveloped, it would bud. Okay? That's the positive sense. The negative sense is essentially the same story, except that it has to come, because it has no AUG on the 5' end, it brings with it the replicase already. And the replicase, once inside the cell, gets going. Why didn't it do that inside of here?
It's not able to.
It doesn't have ribosomes, it doesn't have the energy, it doesn't have the building blocks. So basically, like everything it needs, you know, that's it. Okay? So, I don't know, I just suddenly thought of an analogy that may or may not work. What if somebody showed up and they had this like recipe for this very complicated thing, and that's all they brought? Haha. So I think, okay, get your stuff off the stove. And I know you wanted those eggs or something else, that's for me, and this complicated thing I'm about to make. All your stuff, and then they just like take off. Like, what just happened here? Right, that's what happens to the cells. So, because they do have their own recipe. Now, you don't know how to make those capsids. That's really weird. And you'll see when we get to our immune system, that's what triggers you. Like, what is that? Very strange looking. I don't make stuff like that. Okay, so it brings its own replicase. It makes a complementary copy. Now I've got my end. All right, fine, this stuff can immediately get translated. And over here, it uses this positive side to make copies of the negative in green. assemble. You know, and you might just say, it all seems so like weird. Why would you do any of this? We have no idea. We don't even know what to do with these guys. Are they alive? What is this? What is it? Seems like a very strange way to go about being. And this, I don't know. Okay, and I agree. I mean, somebody who came over here the other day and they really wanted to like, give me another word, Tia. You were like, give me some other word. What can I use? Like, I don't have one. We're stuck. Living, dead, non-living. That's all I got. Okay, so like, the stapler is not scheming to how to make copies of itself. It's like, right? It has no desires this way, but the virus does. So there's some drive in there that we can't quite get our handle on. All right, so we just remind ourselves that protein synthesis is goes in this manner. We've seen this before. We said that this is the the name just went out of my head. This DNA to RNA goes to protein. It's the thing we all say about cells. This is how life goes. And so this first arrow transcription, the second one translation. But we know that for RNA viruses, they need copies of the RNA, and the cell doesn't do that. Okay. So viruses, of course, have to use ribosomes, those are yours, for their protein expression, and they mature in ways that we have no idea, okay, how it is that okay, here's the, I mean, this is a nice picture, but we have never, like, seen how this is really happening. I mean, we take time lapse, but it's not really explaining what's going on. They do mature on their own. And the release, they will bud, and this is a real picture, okay, of the budding. And remember, it's 3D, so it's going in all directions, right? Budding off, taking some of that plasma membrane, or bursting. And that destroys the cell. There's no way around that. Okay. So that is that. But now I want to go to what is